Hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my new beginning series. I'm teaching biotechnology and its applications. So probably today is the last lecture of biotechnology. So we're coming to the end of biotechnology era, you could say, because for the past 3 weeks I've been dealing only about this term biotechnology. So today we are towards the end of the chapter. Hi W, so thank you so much for joining my lecture today. So a very happy good evening, and I hope you're doing good. You had a very good weekend. So please do like, share and subscribe. So thank you so much for joining my lecture once again. Okay, so today we are gonna almost finish off biotechnology and its applications. So in the previous lecture, that was on Friday, I was talking about how biotechnology is used in the field of medicine. So I gave you examples, or I told you how it is being used in the production of recombinant insulin, how it is used in the process of gene therapy, and how it is also used in molecular diagnostics like your PCR, your molecular probe, like your ELISA, etc. So today I'm going to take you to a whole new topic, that is transgenic animals. So I already told you transgenic means genetically modified. So we have genetically modified animals. We have a lot of them. We don't have one or two, we have a lot of them. So why do we make transgenic mouse? So why do we make transgenic animals? So what are we going to gain from them? So that is what is the entire talk for today. And with that, we'll be probably saying goodbye to biotech and W, as you had wished and as you had requested. So you wanted me to teach biology, so from Wednesday I'll be starting, you requested me to teach evolution. So from Wednesday I'll be teaching you evolution, and Friday we have a Menti quiz on the topic biotechnology and its applications. So please do join for the Menti. I'll make sure the community post is there so that you keep a reminder to come on Saturdays 4:15 Menti. Alright, yes? Right. Okay, W. So now we move forward. So we are going to talk about biotechnology and its applications. So it is almost part 5 today. So a quick recap of what I was teaching you in the last class. So in the last class, I taught you how molecular diagnostic works, right? So we were talking about the application of biotechnology in the field of medicine. So under this, we spoke about how insulin production happens. We also spoke about how gene therapy happens. But in the last lecture, we spoke about how biotechnology or recombinant DNA technology is used in the process of your molecular diagnostics. So under this, we spoke about 3 things. So first was your ELISA technique, and then you have your PCR, and number 3, we have your molecular diagnostics. So where, if you remember, sorry, not molecular diagnostics, sorry, we spoke about probe technique, right? So these were the 3 things that we spoke under molecular diagnostics. So let's talk about ELISA. So ELISA stands for enzyme-linked immunosorbent assay. So this is used to detect the presence of HIV or any kind of virus for that matter. Yes ma'am, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Okay, so this is ELISA, so enzyme linked immunosorbent assay. So for example, I have a patient A and I'm suspecting that he might have the HIV virus. So then I prescribe ELISA test to that patient. So what is exactly done in the ELISA test? So I have a rectangular plate which has holes like this. So like this, I have 96 holes. It is 12 into 8 dimension. So I have totally 96. So I don't call them as holes, I call them as wells. So I call this plate as 96 well plate. So on these holes or on these wells, I add the blood of the patient. So it can be either the blood or the serum I can add, right? So when I add the blood, so on top of it, I will add primary antibody. If at all this blood has the HIV virus, that is my antigen. right? So if at all the blood has the antigen, the primary antibody I am adding will go and bind to the antigen. Yes, right. So my antigen and my antibody will bind. Now from the top I will add secondary antibody conjugated with an enzyme. So the secondary antibody will attach to the primary antibody, which is attached to the antigen. So it will be like this: antigen, primary antibody, to which my secondary antibody attaches, conjugated with the enzyme. But I cannot see any of these, right? So from top I will give a substrate. So this substrate, when it is catalyzed by the enzyme, would give me a colored product. So all the wells will turn into a color. So it will be easy for me to identify if the patient has HIV or not. So I will measure the intensity of color in spectrophotometer. So this is ELISA. So the intensity of color is directly proportional to the amount of virus that is present in a person's sample, right? So then we went on to talk about a PCR technique. So I guess PCR is not new to you and you already know much about PCR. So PCR is polymerase chain reaction. Right? So this PCR is used in the process of gene amplification. For example, if I have to amplify a single portion of the gene, I do PCR. So if this is the portion of the gene that I want to amplify, I will do PCR. So the first step being denaturation. So I do need denaturation at a pretty high temperature of 90 to 92 degrees Celsius, right? So I denature the two strands like this, and at this time the primer comes and binds to the two strands. So when the primer comes and binds, that is called as annealing. So I have denaturation. So after denaturation I have annealing. So what happens in annealing is my primer comes and binds to the place where I want the amplification to be done. Yes, you're absolutely right. So denaturation, annealing and extension. So these are my primers. Right? Okay. So once annealing is done, then I do extension step. So annealing is done at a pretty less temperature of 56 degrees or so, and extension is done at a higher temperature of 72 degrees. So here what happens, I have a polymerase that comes and extends this piece, right? So one small portion is now amplified. So here I use the enzyme called as Stag polymerase, which is isolated from a bacteria called as Thermus aquaticus. So what are the requirements of PCR? So first is I will need a template stand to be amplified. So I will need the corresponding primers. So I will need Stag polymerase. I will also need my nucleotides so that it can do the extension. So that is nucleotides, DNTPs, so which includes your DATP, so it includes your GTP, GCTP and DTTP. Adenine, guanine, cytosine and thymine triphosphates I need. So this is what happens in your PCR. So the last molecular diagnostics that we spoke about is the probe technique. So what do? I take the person's sample, I take the person's DNA or the sample DNA. So this is the DNA. I denature the DNA so that it separates it into two strands. So to this particular piece of DNA, I have probes which are already synthesized. So I add probes which are radio-labeled. So I add radio-labeled probes to these sequences. So the probes have complementary sequences to this region of the DNA. I take it from my human genome project. So it goes and binds like this. If this is the entire sequence, so the probe grows and binds like this and it is radio-labeled. right? So the portion where it is not bound, I find out that there is a mutation, and where the probe is bind, that portion is pretty safe and there is no mutation. Because I prepared the sequence from a human genome project, if there is a mutation I will not know, right? So wherever the probe is not binding, so that is where your mutation has happened. So this is also one type of diagnostic technique, and this is what we were discussing in the previous lecture. So now going to transgenic animals. So what are transgenic animals? So animals that have their DNA manipulated to possess and express an extra gene is called as a transgenic animal. Okay, so before we go about transgenic animals, so I have news for you. So I promised you that we are going to have chemistry classes, right? So the chemistry classes will begin very soon, as I promised. So I would like to introduce you the teachers who will teach us chemistry. So we have sir with us who will tell you what he's going to teach and from when he's going to start. So it'll be like kind of introduction. So the sir might come instantly to this session and give you a hi hello introduction, so that you can get used to him and you can have, or you can use his lectures to read chemistry. Alright, so for the past 1 year, students have been asking me for a good content of chemistry in English, which it took me a lot of efforts to come here, and it was tough for me to make a team out of at the back end to come here and deliver the lecture. So anytime during the lecture, sir will come in and say that he's going to teach chemistry, so he'll just introduce. So that is what we would like to do. So please be ready for your chemistry sir to come anytime. Okay, right. So I guess his class will be starting from Thursday. So probably he might come in today's lecture as well as in Wednesday's lecture. He'll just come for 2-3 minutes and say hi, hello. He'll introduce himself and he'll say which topic he's going to teach. So you can mark the time or this set a reminder so that you can come and watch the chemistry lecture. Alright, yes. So continuing about transgenic animals, what are we talking about? So animals that have their DNA manipulated to possess and express an extra gene is a transgenic animal. Suppose, say for example, I have a cat, so the cat is normal. Now I insert a gene inside the cat so that its fur looks like a zebra. So the animal's genome is modified, right? It is not the original one. So now it is called as a transgenic animal. Alright, so now it is called as a transgenic animal. So moving further, so today in the current world, so you have transgenic rats, so you have transgenic pigs, you have transgenic sheep, you have transgenic cow, and you have transgenic fish also produced. So we have so many transgenic animals here. Right? So you have rats, you have pigs, you have sheep, you have cows, and you have fish being produced. But majority of them, that is 95% of all the existing transgenic animals, are your mice or your rat. So we use these animals for our lab experiments. So that is why these are in majority, or these are in large numbers, outstanding the other transgenic animals. But why are these animals being produced? So what gain will I have if I put a zebra fur gene inside a cat? Yeah, of course the cat will express the stripes like zebra. So what is so great in it? I'm wasting my resources, I'm wasting my efforts, right? So then in that case, how will I do something good for the environment? Cucumber is not transgenic. Today we have transgenic cucumber also. So half the time we do not know what kind of vegetable are we eating. Are we eating a transgenic vegetable or are we eating original vegetable? We do not know, right? So, but why, with what motive I am producing these transgenic animals? So when I say you have transgenic cats, you have pigs, you have sheep, you have cows, and you have fish, why am I producing it? I am not wasting my time. The resources are pretty expensive, right? How can we benefit from such modifications, or what is the modification? Yeah, this is a cute mice, right? So let's see how they are being used. So how are they produced first? How is a transgenic mice produced? For example, I take the sperm, okay, and I have the egg. So it is about to fertilize. It is about to fertilize and form a cat, say for example. But if I want to insert a gene, I will insert it inside the sperm prior to fertilization, so that after fertilization, it expresses the gene I want it to express. Okay, so this is probably one way that I help it get into transgen. Mice and rat are the same? No, mice is the small one, rat is the older one, the grown one, right? So this is one way like how I can produce a transgenic animal. The other way is after fertilization has happened, but after fertilization and between implantation there is a stage. So I hope you know after reading human reproduction, if you have read that chapter. So first your zygote is formed, your zygote undergoes a series of division, and after certain division it gets implanted, right? So the stage between implantation and after the formation of zygote is very, very crucial. So in that stage also I can insert my gene of interest, so that is where all your stem cells are there. So that is also the stage where I can insert my gene of interest, alright? Yes? Fine. So this is how a transgenic animal is produced. So why are the transgenic animals are produced? So what is their use? So first is to study the normal physiology and development. For example, if I want to know the function of a particular gene X, so how will I learn the function? So if you want to know the worth of my class, so one day if I stop teaching, you will know. So you will know how worth ma'am's classes were. So one day if your mom goes out, you know how much important your mom is, how much work she does, right? So how gene regulation affects the normal body functions and development. So that is the second one. So that is also why we take transgenic animals. So for example, to study what is the function of a particular hormone inside your body, we use a transgenic mice, because I cannot use W as an experimental model to study something, right? So I cannot harm him. So in that case, we use transgenic mice, okay? So this is the number one reason why we use transgenic mice. So number two, study of diseases. So increase our understanding of how genes contribute to the development of diseases. For example, say a person is suffering from cancer, I cannot study from that person, right? So for example, I can cause the cancer growth, I can introduce the cancer causing cells inside the mice and see what are the complications that it has and which gene gets activated, which gene gets inactivated. So I'll be able to treat the humans who are actually suffering from that diseases, right? So for cancer, we need a model. For cystic fibrosis, where everything gets damaged in your body, you need a model. For rheumatoid arthritis, which is an autoimmune disease, for Alzheimer's, you need a model to study how consequences and complications can happen, right? So number three, to produce biological products also we produce transgenic organism. So we produce useful biological products created by the introduction or portion of the DNA or genes. For example, we have already created human protein alpha-1 antitrypsin to treat emphysema. For example, I took a mice and I introduced this human gene inside it, and the gene is expressed, so I take the protein just like I did for insulin. But insulin I did it in a bacteria. But this alpha-1 antitrypsin I did it in a mice and I extracted the product, right? And similar attempts were done for your phenylketonuria and cystic fibrosis as well. So one more, so these are the list of recombinant proteins that are being produced today in the diagnostic world or in the healthcare world, alright. Apart from that, you also have transgenic animals to give you solid commercial products. For example, a cow was genetically modified and we named the cow as Rosie. So what is so special about Rosie is that it produced a human protein enriched milk. So cow has milk protein, milk has milk protein, but in this cow we inserted a gene so that the human protein is produced in a higher quality of 2.4 grams per liter. Rosie is a cow. Rosie is a transgenic cow. Alright. Hi Prince. Hello. Hi. So thank you for joining my session today. So please do like, share and subscribe. So I hope you're doing well, Prince. Right. So Rosie is a transgenic cow. So we modified her genome so that she gives human protein enriched milk. So in the ratio 2.4 grams per liter. Okay. So milk contained this human protein called as alpha-lactalbumin, which was important for us. So the balanced product for human babies than the natural cow milk. So for example, there are some babies which are born out of C-section, that is cesarean section. So it is generally said that people who are the babies which are born out of C-section are generally less immune. Because when the child comes out through the normal birth canal, so in the birth canal on either sides, you have a lot of microbes and it gives you a lot of immunity to the baby. But when it is pulled up from the C-section, so it has less immunity and there is a probability that the baby can fall sick. So in that case, you have to give a balanced protein or foods that it can enhance or boost the immune system, right? So in that case, if you give the milk of this transgenic cow, the child will have good amount of protein entering inside its body. So that is the main reason we have made this transgenic cow. So this is 𝗏𝐴, a transgenic cow. So she can give milk which has human enriched protein. So 2.4 grams per liter. So the first use of transgenic animal was to study the normal physiology and the development of disease. And number 2, it can act as a model. Right, model for disease. So first is normal physiology and to study the genes. Number 2, to act as model, and number 3 is to produce biological products like alpha antitrypsin and your 𝗏𝐴 the cow. Right? Hi Shweta, yes, hello, I'm good, how are you? So after a very long time you remembered me and you came to my session, so thank you so much. I hope you're doing well, and please don't forget to like, share and subscribe. Alright, so the last reason why we produce transgenic animal is vaccine safety. Yes? Yes, so W is astonished by the cow that we created out of recombinant DNA technology. Fine. So the last reason why we produce transgenic animal is vaccine safety. Not only vaccine safety, any type of medicine, any type of injection is first tested on these animals. So for example, if I create a tablet in my lab, so I will not have the gut and glory to test it on myself or to give it to W, right? So that will become a serious case also later if something happens to either of us, right? So in that case, whatever medicines, whatever cosmetic, whatever drops or ointments comes, it is first tested on these animals. So you would have heard when we were talking about the pandemic, when we were amidst the pandemic, so we saw the growth of the vaccines, that people said the vaccines are under animal trial or under human trial. So whichever vaccine it is, it has to be first given to the animals and tried to them, and then later it comes to the humans, alright. So first is vaccine safety. So transgenic mice is developed for use in testing the safety of vaccines before it's used in humans. So polio vaccine was first tested in a transgenic mice and then it came to the market. So whichever drug you have today in the market has taken 10 long years to do all the testing and come for the commercial service in the market. If successful, they replace, so if the mice does not have any side effects, if the mice is safe, then we go to monkeys. And the final thing is we give it to humans. So giving this to monkeys is still fine, but we also do cosmetic testing in the transgenic animals. That is chemical safety testing. So any new lotion, any new products, cosmetic products, any lipstick, anything, that kajal, anything that comes in the market, it is first tested on these animals for the toxicity level. For example, if I put a lotion on my skin and if my skin burns, that is a very serious issue, right? So to avoid all this, they first test the cosmetics or any kind of chemicals on animals. So imagine if I have an injection for my eye cancer, that is retinoblastoma. So if I come up with an injection to help the retinoblastoma patients, the patients with eye cancer, that will be first treated to animals. So poor animals, I will make them have the retinoblastoma grow. So I will inject the tumor cells so they have this eye cancer. Then I will inject it into their eyes. Okay. Vaccine research time, many people and animals die. Yes. For example, initial trial, I am making a vaccine. So this is the first time I have made a vaccine, but I am sure it will help treat one disease. Probably in the first round, when I gave it to 30 mice and 30 mice died, so I have to recheck my formulation. But yes, the mice that is dead. So here we are evaluating human value more compared to these poor creatures. Okay. Yes. So transgenic animals carry genes which makes them more sensitive to toxic substances than the non-transgenic animals. So for example, if I am testing a lotion, I put the chemical sensitive gene into the mice, into the transgenic mice, and then I do my testing. So then the toxic substances are exposed. Yes, it is really sad that we are using these animals, but do we have an option? No. I cannot directly test a drug on the ones that whom I love, right? So I have to take a chance, I have to risk, I have to sacrifice one thing to save the other, right? But you can ask me, ma'am, we are killing so many animals. Is it not an ethical issue? How can I just like that kill an animal? So let me tell you, so if you want to do animal testing, it is not easy. So if I have to do animal testing, I cannot go to some gutter, pick up rats and come and do the testing. No. So there's a particular committee called as the Genetic Engineering Approval Committee. So I have to write a proposal to that committee stating, this is my project, this is what I'm trying to find in my project, this is what I'm trying to test, and for that I need so many animals. Only if they approve, they will send the animals, because transgenic animals are produced in a commercial lab. Okay, so they will send animals to you if your project is practical enough or valid enough. So by chance, imagine I am a scientist, I am working with 30 mice, and by handling error, suppose if I have to inject something into the neck of the mice, and by handling error I kill the mice, I am given a warning to stop working in lab for 6 months. If by chance 2 animals die because of my handling error, I am debarred from lab for 1 year. So yes, there is a genetic approval committee to whom I have to report. Suppose if they give me 50 animals, I have to send the report of all the 50 animals back to them. And suppose if I have killed a particular animal because my medicine, that is what is the end product of my medicine. For example, I am studying a particular cancer, so I have grown the cancer into the mice. So at the end, the mice will die. But how do I dispose the mice? I cannot just throw the mice, because I can study HIV virus on the mice, right? So I have to dispose the mice properly. I have to incinerate it. I have to burn it. And that certificate also I have to submit it to the committee. Only then my research work will be approved, right? There is something called as biopiracy after this. So use of bioresources by MNCs and other organizations without the proper authorization from countries and people concerned without compensatory payment. So what do I mean? For example, I hope you know what is turmeric. Turmeric is haldi. So turmeric is native to India. And this turmeric has a compound called as curcumin. So this compound is very important. So since Indians use turmeric in our diet, we do not get Alzheimer's very easily. So Alzheimer's is prevented because of the curcumin that is present in your turmeric. But before we could realize this, the foreign countries understood this, and they took the turmeric plantation from India and they started growing it there. They didn't stop with that. They said, now without our permission, you cannot grow turmeric. So that was so unfair in the game, right? So it actually belonged to our country, but you took it from us without even our knowledge. And now you're saying, I cannot grow it without your knowledge. So that is called biopiracy. You tend to cheat the original company. So a lot of farmers do not know anything. If some foreigner comes and they ask, they can give. That is the nature of farmers, right? But these companies cheat people like this, and that is called as biopiracy. The same thing happened with neem also. So neem also is native to India, but knowing its medicinal value, they took it abroad and they never gave us a compensatory payment also. So today if I have to have a turmeric farm, we have to take. So we fought for it and today we don't have to do it. So that is biopiracy. So the last one is bio-war. So I guess you know what is bio-war and bio-weapon. So people say your coronavirus is bio-war, bio-weapon. I don't know. So bioweapons, for example, you have anthrax virus. Anthrax is a bacteria. It causes a serious disease in the cattle by which the cattle dies. So if I spread one anthrax in one cow, it quickly spreads to all the cows and I kill it. So it is like a mass killing, so it is called as a bioweapon, right? So these are the things that is there. Foxnut only in India. Walnut, which country? Walnut comes from the Western countries, like probably Italy or US, I don't know, I'm not sure. So, but it is from the West, okay? So this is all about biotechnology. So I guess I have explained biotechnology in bits and pieces but fully. So on Wednesday we are starting with evolution. On Friday we have a quiz based on this chapter, biotechnology and applications. So 25 questions, 25 seconds for each question. So join me on Friday for the mentee, but before that, Wednesday at 4:15 I am teaching you evolution. So please join me for the same. Alright. So with this we end today's lecture. Yes, so if you genuinely liked my way of teaching, so please do like, share and subscribe. So thank you so much for joining my lecture today. So probably he's coming on Wednesday. It's a surprise for all of us. Alright, so he said he will come. So if he's not coming, it means he'll come on Wednesday. So I mentioned it, but probably you'll have the class from Thursday, I believe. So please be there. Yes, we are gonna start chemistry in this channel as well in complete English. So it will start from Thursday. So please be ready. Alright, so thank you so much for joining my lecture today. So please do like, share and subscribe. So thank you so much once again. Have a great day. Thank you and bye.